shall we go to part 2 and get into more chemical immunotherapeutic approaches. So what I talked about up to this point is kind of to give you more of a background of the general field of immunotherapy and how the host immune system can interact and recognize cancer potentially and when this is able to escape. And a lot of the research is done in immunotherapy is really involved biologic type molecules, right? So therapeutic cells and antibodies, large proteins. And what our lab is really focusing on, and what we're contributing to the arsenal of immunotherapies, is really in the area of smaller synthetic molecules. So that's really what we're going to talk about at this stage in the lecture. What I've shown here is really a schematic of these little squiggly lines and triangles and hexagons that are able to bridge or recruit immune machinery that's already inside of the host in the serum or surrounding the tumor, and actually increase its target residence time, increase the probability that the immune machinery is going to actually kill the cancer cell. Okay, so using these small molecule approaches. I want to give you a brief history of where we're starting from here. Is really this idea of a bifunctional molecule that's able to recruit endogenous antibodies that are not specific for cancer, but use them in a manner that we can redirect immune machinery that's already inside the host against the cancer. Okay? So these molecules, as I would classify as a non-covalent dimerizer strategy, where you can see this molecule here with a small molecule on one side that binds, here's a hapten that binds an endogenous antibody, and another small molecule that binds a tumor receptor, akin to the first slide in this presentation. The idea is that these bifunctional compounds kind of behave like antibodies, although much smaller molecular weight. And the idea is that it decorates the cancer cell that overexpresses a receptor, as long as the small molecule that you have is selective for binding that receptor. And then the hapten that's exposed, sorry, I keep losing this mouse on this white background, the small hapten here binds to endogenous antibodies. So one such hapten and one such endogenous antibody that we have well characterized is endogenous antibodies against this DNP molecule, okay, this electron-deficient arrow ring, dinitrophenol. Okay? You tack this small molecule onto one side, acting as the hapten, you can recruit antibodies that are already in the serum of humans that will bind this and then recruit them to the target cell. And depending on the isotype, you can recruit immune effector cells like natural killer cells through those Fc gamma R3 interactions with the Fc domain. And then through recruiting antibodies and immune effector cells, you can actually achieve targeted cell death. Okay, so that's the idea. The small molecules act similar to antibodies. So in a way, you could think of it as instead of introducing a monoclonal antibody therapeutic, the compound can recruit high levels of antibodies that are already in your serum. Now, why they are depends on the antibody that you're actually going after. That's a whole other topic of conversation. But the idea is that you can recruit these antibodies, and you can program them to have quite a high affinity and selectivity for a receptor that's overexpressed on a cancer cell. Okay? Another neat thing about these molecules is that because they're synthetic, they have the potential to engage in rapid turnover events against the cancer cell. So once you get phagocytosis, let's say, of the cancer cell, proteases are not going to touch the molecule, whereas a biologic, the protease can actually chop it apart. So that antibody, for example, may no longer be active after one event. Okay? Of course, you're not administering a high concentration of antibodies ex vivo. That's required to get a therapeutic concentration because you're harnessing antibodies that are already inside. So this kind of presents an administrative type advantage. All right. It's also important to understand from the perspective of chemotaxis, because these are much smaller than cells, we're no longer relying on chemotaxis to recruit the effector machinery as much as we were in the case of T cells, for example, whereas now the antibodies kind of operate through a passive diffusion and then get localized to the target where they make the high affinity binding interaction. So it's a little bit of a different framework conceptually in how we get targeting. Okay, it's more of a passive targeting, if you will, as opposed to a more active one using chemotaxis. So I just want to take you through a little bit of the kind of what the chemist really thinks about in this arena. Is you start off with a target receptor. So in the case of these antibody recruiting molecules against cancer, we went after urokinase plasminogen activator receptors. So the idea is that you know the receptor, you know a binding site on the receptor where you can take a small molecule, as shown here, this anthraquinone type isoxazole core that has affinity for UPAR, okay, at a certain binding site. And you can tether this to the hapten that binds antibodies, okay, using synthetic chemistry to get your bifunctional molecule. All right? And the idea is that you can use computation to basically try and understand in silico how this molecule will interact with this protein and make sure that it can interact in such a way that it engages in interactions with amino acids in the binding site that are important for binding affinity to the target cancer receptor, and at the same time keep this antibody binding terminus that's actually the hapten that the antibody will recognize solvent exposed, so that the antibody can also bind this molecule, okay, which is important so that we can get a high valency of antibody around the cancer cell, okay, and then potential immune effector function. So this here is basically showing you a ball and stick version of this molecule on the right, this antibody recruiting molecule, now docked, so using computational algorithms and software, how this molecule is predicted to behave when it binds this compound, okay? Binds this receptor, sorry. So UPAR is one such, I should mention UPAR is one example of a receptor that's very overexpressed on the surface of certain cancer cell types, especially the highly metastatic, glioblastomas, cancers of that nature, okay, that require rapid metastasis and shedding of the extracellular matrix. UPAR is involved in interactions with a protease urokinase, which some of you may be familiar with. So the basic way that we validate a bifunctional compound that's able to bind antibodies and a cancer cell receptor on the surface of live cancer cells is thanks to the work of immunologists, really through established flow cytometry assays. So using fluorescence, we can actually watch how these molecules, these bifunctional compounds, shown again by this triangle and small hexagon, can actually bridge antibodies that are in the serum, for example, to bind the surface of a cancer cell. Shown here, A172 glioblastomas that overexpress UPAR. Okay? And then again, using streptavidin biotinylation, we can actually image or visualize the formation of what we call a ternary complex between this bifunctional compound, the anti-DMP antibody that binds that dinitrophenol hapten, okay, and UPAR, shown here by these Ys on the surface of a cancer cell. And because we're bringing fluorescence to the cancer cell, we can actually monitor how well these molecules bind the cancer cell and can recruit antibodies. To say it another way, how well these molecules form ternary complexes with cancer cells. So you can see here, as we get more fluorescence, the cell population shifts to the right in a UPAR binding dependent manner, because the fact that we see a shift in the cell population fluorescence means we must have molecules that binds UPAR on the cancer cells and the fluorescently labeled antibodies. Okay, so after making a bunch of different compound variants that have different binding affinities for UPAR, we can evaluate their ability to bind UPAR on the cell, okay, and also the anti-DMP antibodies. And what we found actually was that there was a correlation between binding affinity for UPAR and ternary complex formation, which makes sense because as tighter it binds UPAR, you would expect the tighter it binds the cancer cell surface and is more present to bind anti-DMP antibodies. Okay? And so you would imagine this ternary complex in vivo would be what's responsible for an effector cell mechanism like phagocytosis or antibody-dependent cellular cytotoxicity. All right? And just what I want to show you here is how there's immunobiological flow cytometry assays where we could actually monitor phagocytosis of cancer cells using these antibody recruiting bifunctional molecules. All right? Using this two-color flow cytometry assay, where the cancer cells are stained with one fluorophore, membrane fluorophore, and the immune effector cells, here we use U937 monocytes as the other. We can monitor the phagocytosis of cancer cells by monocytic effector cells by the merging of these two colors in flow cytometry. Okay, and what you can plot out is this histogram of phagocytosis and with a dose-dependent response of the antibody recruiting compound. Okay? So what we see here is in a dose-dependent manner of increasing the concentration of the antibody recruiting compound, we're getting an increase in the phagocytosis of cancer cells through this ternary complex mechanism. Okay? Of course, when you're performing an assay like this, there's certain controls you want to perform to make sure that the phagocytosis is indeed dependent on the molecule binding cell surface UPAR. So we would, of course, include competition experiments with exogenous UPAR to knock out phagocytosis. You can see a control there. The addition of a molecule that can bind UPAR but cannot bind the DMP antibodies. You can see here that this has this pyrimidine molecule instead of a dinitrophenol. This would act as a competitor for ternary complex because it can't bind the antibody, sorry. So these types of controls, what I want to draw your attention to is that although we have efficacy against cancer cells, we get this unique bell-shaped distribution of efficacy versus concentration. So we see here that we reach a plateau at some concentration of small bifunctional molecule followed by a decline in its efficacy. So that's one feature of this bell-shaped curve. The second feature I want to point your attention to is the fact that we're only getting 40% phagocytosis. Why are we not getting 100? Okay? Now it's interesting to note that these compounds behave like antibodies, and it turns out that this type of phenomenon is actually very relevant to monoclonal antibodies as well. They also have these bell-shaped signatures. This is actually, I should mention, bell-shaped signatures known as the Prozone effect are a well-known phenomenon in pharmacology, not necessarily to organic chemists. And so, we were quite interested to see these bell-shaped curves where a pharmacologist might have looked at it and been like, of course, right? So, something interesting to note. What I'd like to also draw your attention to is the fact that these compounds in B16 allograft models could significantly reduce the rate of tumor progression. Okay, in mice that we immunized to form high levels of DMP antibodies, because the mice don't contain the antibodies the way that humans do. With B16 melanoma tumors that we grafted human UPAR onto, we could see that when you treat with just PBS, we get this rate of tumor volume increase with time, but in the presence of the antibody recruiting compounds, it was markedly slower, actually akin to doxorubicin, a type of chemotherapeutic. And you can see here these Kaplan-Meier survival curves. The antibody recruiting compounds actually bestowed a substantial increase in their survival. And unlike chemotherapeutics like doxorubicin, without any weight loss, you can see here that if anything, the mice kind of gained a little bit of weight when they were on antibody recruiting compounds as opposed to doxorubicin, kind of highlighting one of the real powers of immunotherapy in that it's very targeted, okay, and that there's the real potential for a lot less off-target toxicity compared to chemotherapies, okay, where it's a lot more non-selective killing. However, after treatment, the tumors do return, so we don't get maximal protection over time. Okay, they do return, and so this really kind of begs the question: well, what's happening here? Why is this not able to actually form an actual cure? Why is it not preventing cancer from relapsing? Okay, and so one of the things that we're thinking about is really, what can we do to make these therapeutics more efficacious? What can we do to try and modify the system so that we don't get this remission or this relapse, I should say. And recall that the antibody-dependent phagocytosis assays actually showed that we got a maximum in phagocytosis relative to what's theoretically possible. Okay, so this kind of stuff has us wondering, what's really happening on the molecular level with monoclonal antibody therapeutics and antibody recruiting compounds, and what can we do as chemists to contribute to this area? So here I have a little schematic with well-known equilibrium arrows for us to think about how these molecules and cells can interact non-covalently from a very simplified perspective. You could imagine an effector cell required to actually phagocytose the cancer cell. However, it requires the antibody, here that we designate B, or the antibody recruiting molecule, to bridge the effector cell with the cancer cell to get these ternary complexes that are responsible for efficacy. Okay, you can see here two equilibrium constants resulting in the ternary complex that's responsible for the death of the cancer cell. Also recall that it's very dependent on the valency of antibodies that are covering the surface. Okay, the actual efficacy of cancer cell death is highly dependent on the valency of antibodies binding the surface. Okay, and so the way we think about it from this kind of thermodynamic cycle is really that you can have either the antibody bind the cancer cell, followed by the immune effector cell, or vice versa. The order isn't really substantial. What is important to note here is that the ternary complex, ABC, that's required for phagocytosis or antibody-dependent cellular cytotoxicity is highly dependent on the concentrations of these species already present in vivo, which you can't easily modify. Okay, the concentrations of the antibodies that you're going after or that you're introducing, it's a very difficult operation to control, as well as the affinities that these antibodies have for their targets, okay, and the immune effector cells in turn for the antibodies. So this kind of has really inspired us to try and get a better understanding of these molecular processes and try and improve these affinities and try and improve how we recruit these immune effector cells to the surface of the cancer cell. Okay? So it's important to note, as you remember the bell-shaped curve that we saw in the phagocytosis profile, that as we increase the concentration of the antibody recruiting molecule, we got that bell-shaped curve. So we reached a plateau in phagocytosis followed by a decrease. Okay? And on the molecular level, what's really happening here is we're getting this bridging compound that can be a monoclonal antibody therapy or an antibody recruiting molecule that forms this bridging species between the cancer cell receptor and this immune effector cell receptor over here, which at the same time partitions between these inactive bifunctional complexes. Okay, so these binary complexes cannot induce phagocytosis or antibody-dependent cellular cytotoxicity. Okay, so this is a large problem with these types of therapeutics, is that you always reach this kind of maximum at this top of the bell, followed by this decrease known as autoinhibition, because you're getting this partitioning. All right, so we think that this is largely responsible for this reduction in the efficacy of monoclonal antibody therapies and ARMs, all right, because of these equilibria. So this is the difficulty, getting the maximum ternary complex without getting these binary complexes that can't actually induce any anti-cancer killing. And actually what's been shown is that looking at actual clinical data using monoclonal antibody therapies, when you fit it to some mathematical models that have recently come out, what we see is that the dose response curve of cytotoxicity against cancer cells with the monoclonal antibody actually reaches a maximum of around 30% of what's theoretically possible, which is very similar to what we saw in the ADCP assays with the ARMs, that we weren't getting anywhere close to 100% efficacy against the cancer cell, okay, followed by this decrease, this bell-shaped decline. You can see that the actual mathematical models are actually predicting what we're seeing experimentally. So this is really interesting. This actually dictates that the monoclonal antibodies are not killing the cancer cell with the efficacy that's theoretically possible. Okay? Neither are antibody recruiting molecules, which you would imagine because they operate through a very similar mechanism of ultimately recruiting immune effector cells like natural killer cells to kill the cancer cell. In this case, you can see here that the max is predicted mathematically by the concentration of C total over this dissociation constant. Okay? This dissociation constant, KBC, is actually affinity, the native affinity between a monoclonal antibody and CD16A, which is a receptor on the natural killer cell. This is not an affinity that we can improve very easily. Okay? And so that basically means that unless you can get this affinity way, way higher, you're always going to have this maximum plateau way under what's theoretically possible, which means you'll never get the valency around the cancer cell that you could get if that affinity was a lot tighter. As well as the concentration of these immune effector cells, which of course in vivo we can't really control when we're administering monoclonal antibody therapies because the natural killer cells that are in the host are already in the host. So we can't really manipulate that concentration of C. So what we're doing right now is we're trying to think about chemical strategies where we can maximize ternary complex on the surface of cancer cells so that we can evaluate what this maximum theoretical efficacy will look like and what this can do to cancer relapse rates. Okay, so that's what we're really working on in the lab right now as we speak. Just to recall here, this ternary complex equilibrium where we get this partitioning between an active ternary complex that's responsible for ADCP or ADCC and inactive species where we have excess concentration of this bridging molecule. So what one of the things that we're really interested in is actually simplifying these multiple reversible binding steps using covalent chemistry. So what the lab is really working on right now, or one big area, is we're developing chemistry that can actually covalently program the antibody that's already present endogenously with tumor binding capabilities. So instead of three things having to come together reversibly with two dissociation constants, now we're actually minimizing that to one, and then eventually to zero as we get dimerization on both ends. And what this is actually predicted to do is to end up resulting in a lot higher density of antibodies binding to the cancer cell surface, a much greater avidity to recruit the immune effector function. Okay, so we're actually in the process of developing chemistry that can site-specifically refunctionalize an endogenous biomolecule, okay, potentially in vivo. So this is actually a pretty powerful idea. So you can imagine here, the bridging molecule now sticks irreversibly to one of these molecules in vivo. Okay, so it's now reprogrammed to bind this target receptor. So in this way, we can think of it as kinetically controlling ternary complex formation, okay, between this bridging species and the cancer cell receptor. And this here brings us back to our thermodynamic cycle, where now we have these irreversible arrows showing that the equilibrium has now been reduced and is much more simple. All right, so we're basically using covalent chemistry to enforce the proximity of immune effector cells with a target cancer cell, as opposed to relying on a more transient reversible binding interaction, as is the case with monoclonal antibodies or antibody recruiting molecules. A little bit about how this chemistry works. Essentially, you want to use a chemical reaction where you have a small molecule ligand. So in the case of antibodies, you would use a hapten for the antibody. So remember DNP antibodies, how they recognize that dinitrophenol residue. What you would have is the DNP actually bind the antibody and basically bring this reactive group in very close proximity to reactive amino acids in close proximity to the binding site. And by affecting this reaction, we can actually permanently label the antibody with a small molecule of interest. In our case, this interest is actually tumor receptor binding ligands, so that the antibody that's endogenous, that before only recognized dinitrophenol, now will actually bind and recognize a cancer cell receptor. Okay, so that's a lot of what we're working on here, and basically trying to tune the kinetics of these reactions so that we can really target the protein of interest, like the endogenous antibody of interest, in the presence of several other proteins. So right now we're going after a number of endogenous antibodies, anti-DNP antibodies that are in serum, as well as a number of carbohydrate-specific antibodies that are already present in us, or by using the actual small molecule ligand that antibody recognizes, we can target chemistry to label that protein in vivo selectively in the presence of other proteins with tumor-targeting small molecules. Okay? I just want to mention that actually we're collaborating with Dr. Jacob McGolan in some of the small molecule synthesis of this folate receptor targeting construct. And some exciting work that's going on in the lab right now with one of our chemical biology co-op students, Nick, who's sitting in the audience. We're already getting some docking data that's actually helping to guide the design of these covalent labeling molecules based on crystal structures that we have of the antibodies. We can design molecules where we can understand how it's going to bind and fit into the binding site of the antibody and direct the chemical reaction specifically close to that binding site on the antibody, okay, with tumor targeting functionality. So there's a lot to consider here from a chemist's point of view in terms of the spacings and the reaction labeling rates that we'll get. You can see here that the docking is predicting this tyrosine in close proximity to this reactive acyl-imidazole carbonate here, which will actually suggest that this tyrosine has a good chance of being covalently labeled with this tumor-targeting folic acid derivative here, which will allow it to bind folate receptor, which is overexpressed on a large number of cancer cells, especially ovarian. Okay? So we just recently transferred this presentation from Keynote to PowerPoint right before, so there might be some things that are a little bit offset. But the idea is that we can actually go after other immune machinery, not just antibodies. We could potentially go after the immune effector cells themselves. So instead of relying on covalently labeling antibodies that then non-covalently recruit immune effector cells, we can go straight to the source and actually develop chemistry that programs the immune effector cell to have tumor targeting function. So using the same kind of chemical technology, we can develop these labeling molecules that binds the CD16, which is an activator receptor on natural killer cells, and covalently label it with tumor-binding small molecules so that this effector cell is now permanently programmed with tumor-binding small molecules. So when it sees a tumor, it's going to bind, it's going to be sequestered in that environment, and has a greater probability of actually killing the cancer cell. Okay? So right now we have some students that are actively looking at designing small molecules that can bind CD16A so that we can turn them into these covalent dimerizers. We can program or decorate the cell with tumor targeting functionality. And shown here is some work that a couple of the other students have been actively doing in the group, Alex Wang and Eden Kapkan, also chemical biology co-op students that have been doing a great job in kind of charging or leading the charge in the development or discovery of these small molecule ligands that bind CD16A on natural killer cells. And shown here is really a docking of a known crystal structure of CD16A with a candidate small molecule here, this benzothiazepine small molecule in the bottom left, which is a candidate molecule to bind CD16A that we can use to target labeling chemistry against this receptor. Okay? And then just shown here is a synthetic scheme of what Alex is actually working on right now to synthesize this small targeting molecule. So I'm just going to finish off the talk with another program, another platform that's ongoing in the lab in collaboration with the Wiley Laboratory in the chemistry department. Is really this idea of designing polymers or oligomers, synthetic molecules that are decorated with small molecule tumor targeting ligands with an optimal display so that we can form high avidity binding interactions with a cancer cell that you would not form with a normal healthy cell because it does not express that concentration or that level of cancer receptor. Okay, so healthy cells, the idea is that healthy cells have a much lower, you guys remember in that first slide we talked a little bit about overexpression of receptors that healthy cells also have but at lower amounts. The idea here is that the polymer is optimized with small molecules that bind the cancer cell receptor of interest so that it binds the cancer cell with high avidity but will not bind the healthy cell because it doesn't have the same array of receptors. And that's how we get tumor targeting selectivity. Once we can achieve this, we can further elaborate the structure of these polymers with small molecules that bind CD16A, as I just showed in the previous slide, and recruit immune effector cell machinery to kill the cancer cell in that manner, okay, and get antibody-dependent cellular cytotoxicity. Now, this is highly relevant, for example, Herceptin, as we had discussed previously, how it's actually able to target breast cancer cells. What I didn't tell you about Herceptin is that it really only works in a very small subset of breast cancers that are called triple positives, so they express this HER2 at very high valencies. Once the valency of the HER2 is diminished, the antibody therapeutic actually is no longer functional against this, okay? And a large reason why we think this is because we're not getting enough antibodies binding to these lower HER2 expressing cancer cells. Keep in mind, this is still overexpressed relative to a normal healthy cell. It's just not as overexpressed as in the most aggressive of these HER2 expressing breast cancers, okay? What we can do with polymers, and what we're excited about, is that we can actually deconvolute the binding to the tumor receptor from the binding to the immune effector cell receptor. You can see here, we can still bind HER2 lower expressors by how we optimize the presentation of these tumor binding ligands. And at the same time, can promote a very high decoration density of immune effector cell binders, which would not normally occur if you're just relying on antibodies. You can see here you'd have two, let's say for example, 2 antibodies binding where the immune effector cells are like, there's nothing wrong with that. But with this, we could actually program it so that the effector cell binding event is actually quite high avidity. Okay, so that's really another area that we're working on to target cancer cells with immune machinery. And so yeah, just to wrap that up, this is really the basic idea of what we're calling avidity-driven cancer cell targeting polymers. So we're using the avidity of the interaction between multiple cancer cell receptors and these optimized polymers to really drive recognition of the cancer cell and recruit immune effector machinery. So with that, I'd like to thank the talented students in the group that we have working right now, Nick, Alex, and Eden, who are all very impressive as third-year co-op students and really driving this program off to a strong start and helping us understand how these molecules interact with the target receptors so we can devise chemistry that's very orthogonal to labeling these receptors. As well as support from the departments and a list of excellent collaborators, Ryan Wiley in chemistry and John Valiant in chemistry, Dr. Singh, Dr. Mogolan in biochemistry, and Dr. Matthew Miller, also at Merck. So thank you very much for having me out here and for your attention.